Okay, well, what I'm going to do, hopefully, is going to show you what I think are some of the astonishing molecular machines that create the living fabric of your body. The way I'm going to show this stuff to you is through digital animation that reconstruct biology derived from the scientific data. Now, what I'm doing is absolutely nothing new. Scientists have always created pictures as part of their thinking and discovery process. They create pictures as part of what they're observing with their eyes, whether it's up through a telescope or down through a microscope, and also what they're thinking about in their minds. I've got a few examples just to illustrate this, and we start, of course, with Richard Fineman, the legendary communicator, the artist, the musician, the dad, who also did some physics, and his diagrams, which he invented, his geometric diagrams to help get through all the massive calculations he was doing. And what I find particularly fascinating about these diagrams is they don't represent physical phenomena. They are representations of mathematical probability functions of particle transition states. And I have to say quite confidently that as a biologist, I have no clue as to what these diagrams represent. But I still admire them for their elegance and their simplicity. On much more comfortable ground for me is Charles Darwin, the superstar of biology. And with this entry in his notebook, he begins in the top left-hand corner with "I think," and then sketches out the first tree of life, which is his picturing of what the connections are between species through evolutionary history, the origin of species through natural selection and divergence from an ancestor population. If I had a tattoo, I'd do that one right there. But I'm not that kind of a guy. Anyway, I used to go to lectures, many lectures by molecular biologists, and I used to find them incomprehensible with all the jargon and technical language and the focus on individual molecules. Until about a dozen years ago, I encountered the artworks by David Goodsell, who is a professor of molecular biology at the Scripps Institute, and he does countless paintings of the molecular world. And what you're looking at here is actually a transection through blood. The yellow-green area is the fluids of blood, which is mostly water, but it's antibodies like Pamela was describing, it's sugars, it's hormones, and so on. The red region is a slice into a red blood cell, and the red molecules are hemoglobin. And hemoglobin are actually red, that's what gives blood your color, and they're like a molecular sponge that soaks up the oxygen in your lungs and carries it to other parts of the body. I was very much inspired by these sorts of images, and everything's to scale, and there's been a lot of work that's been put into these sorts of images. And I wanted to see if I could recreate or redo this with digital animation. And the main reason I wanted to try this was digital animation can show dynamic behaviors. It can show active systems, which is a fundamental feature of biology. So if we could set up the first video, please. Okay, you're looking down on the surface of a stem cell. This view wasn't working for me. It's magnified about 1 million times. So I rotated the camera down, and this blue structure is like a sensor. It's a receptor, and it scans the bloodstream for a particular message, a hormone, and that's the hormone coming in and docking there, the yellow object. But anyway, in this particular case, it turns out you need 2 of these receptors, both with their hormones docked to get together. And when they do, the signal is transmitted into the stem cell, and it creates lots of blood cells. And this discovery of this particular hormone, colony stimulating factor, has enabled millions of people to survive high-dose chemotherapy. If we could have the next video, please. For the rest of my talk, I'm going to focus on DNA and DNA science and show you some visualizations of that. So here is DNA in its classic double helix form. And if we unwind the double helix and unzip it, you start to see its structure. These things that look like teeth that are wiggling away there, those are the letters of genetic code. That's where the genes are written. And this stuff is kind of really important, and if you don't believe me, you can ask Craig Venter at a coffee break. But I want to talk about a different part of it, which is these 2 strands, and they physically run in opposite directions. One strand runs in one direction, one strand runs in the other direction, for reasons I can't go into now, but it creates a number of complications for your cells, as you're about to see, most particularly when the DNA is being copied. And what I'm about to show you is an accurate representation of the actual DNA replication machine as it's occurring inside your body, at least at 2002 when this was created. The DNA is entering the production line from the left-hand side. It hits this collection, this miniature biochemical machines. The blue donut-shaped molecule is splitting the DNA strands. One strand can be copied directly and can be seen being copied off to the right-hand side. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time. The end result is 2 new DNA molecules. You have billions of this machine operating approximately at this speed, right now whirring away with extreme accuracy, copying your DNA. What I'm going to show next, this was work from a long time ago. I want to show you some of my current works in progress, and this is the first time I've ever presented to anyone, so be gentle with me. It will show you some updated technologies, but also updated science, and I'm trying to tackle even bigger whopper protein structures. So again, we begin with DNA, and it's wiggling and jiggling there because of the surrounding soup of molecules, which I've stripped away so you can actually see something. DNA is about 2 nanometers across. That's really quite tiny, but in a typical human cell, the average DNA strand is about 30 to 40 million nanometers long. So to keep it organized and to regulate access to the genetic code, it's wrapped around these purple proteins called histones. The name's not important, but it's sort of spooled around these and packaged up and bundled up. All of this field of view is a single strand of DNA, and this huge package of DNA is called a chromosome. And we're going to focus on chromosomes. We'll come back to them in a bit of a minute. The camera is now zooming out, pulling away from a single chromosome. We're now zooming outside. That donut-shaped structure is a nuclear pore. It's a gateway to this compartment that holds all the DNA. And we have a whole semester's worth of biology right in front of you, and 3 minutes to go, so to just focus on the chromosomes. We're pulling out to a view which is as if you would look down a normal microscope. This is a living cell. It's under time-lapse, so that's how you can see it all moving. The nuclear envelope breaks down, the chromosomes are released, and they go through this very striking sort of movement, which just seems to be all focused on, I've added these little red dots, they seem to be tugged and pulled to this equator position. And when the cell feels everything is ready to go, the chromosomes are ripped apart. One set of DNA goes to one side, the other set of DNA goes to the other, identical copies of DNA, and the cell then splits down the middle, creating 2 living cells, 2 daughter cells. So we're going to rewind and just focus on the DNA, the chromosome, and look at its structure. So again, here is that equator moment. It's the chromosomes are wiggling around, and I'm just going to isolate just one chromosome out of it, and we're going to have a look at its ultrastructure and how it's made. So here's one chromosome. Those things that are sticking out either side that look like whiskers are part of the cell's scaffolding. They're called microtubules, but they're for orienting the cell and for locating it, that center origin point. The sausages are, it's one chromosome. It's got 2 pieces of DNA. One's wrapped up in one sausage, the other one's the other strand of DNA. The red region is what we're going to focus on. It's a gigantic, huge protein complex which is made up of 100 to 200 proteins, and it is the interface between the chromosome and these long fibers, these microtubules. We've known about this red structure, I've labeled it red, for more than 100 years, and we have a lot of information about it. It is obviously central to the movement. We have no clue, really, as to how it's actually achieving the movement, but it's also a broadcasting system. It tells the cell that it's not ready. It's also a tension sensing system. It's able to tell through tension whether it is ready to undergo the separating of the chromosome. And it also is able to, it grows these microtubules, or it hangs on to the growing and shrinking microtubules. They're growing only in this particular image. So it's able to transiently couple onto the microtubules. It's turning green because it's sensed that the tension is just right. All the proteins get ready, and you'll see there's one little last bit that's still remaining red, that stop signal, and that is the signal broadcasting system. And it's literally walked away down the microtubules and away from the kinetochore. I mean, it's that mechanical. And so with a little bit of eye candy, molecular eye candy, these little guys are kinesins, are like little courier molecules carrying those little packages on their back. And here are the dynein coming from the kinetochore carrying that red broadcasting system. And they've got their long legs so they can step around obstacles where the other guys, they could. So the kinesin can only walk one way, dynein can only walk the other. So purpose of my talk is to really give you guys some feel for what molecular biology is actually like, and perhaps to woo some of you nanotechnologists and physicists over to the hot tub of biology. Because I have to say, the water is warm and it's bubbling. So come on over. Thank you.